So flow cytometry is a powerful analytical technique that is used to measure physical and chemical characteristics of individual cells or particles that are in suspension. Some principles, so we have laser-based detection. These cells are suspended in fluid and passed one at a time through focused lasered beams. For scattering and fluorescence, so as each cell passes the laser, it scatters light and may emit fluorescence if labeled with the fluorescent markers. For detectors, it'll measure the forward scatter, which is related to the cell size, and the side scatter, which is related to the cell granularity or internal complexity. For the fluorescence intensity, for detecting tagged surface or intracellular markers. So how it works, we have cells that interact with the lasers. The fluorescent tags on the antibodies, like a CD marker, will emit specific wavelengths. Detectors will capture light signals to quantify and classify the cells. Some benefits of this technology is it is rapid, quantitative, and highly specific, and it'll allow for analysis of thousands of cells per second. Flow cytometry has many uses. One of them in the clinical lab is immunophenotyping. So this would help to identify and quantify different immune cell populations. It's essential in diagnosing hematological malignancies, especially leukemia and lymphoma. It can also do cell sorting, so it can separate and collect specific cell populations for further analysis or even for therapeutic use. For another assay, we could do a functional assay, and it assesses cell activation, cytokine production, or apoptosis. For minimal residual disease detection, it detects small numbers of malignant cells post-treatment, which is crucial for patient prognosis. It can also look at stem cell analysis, HLA typing, and transplant monitoring. So how to interpret the dot plot? So we have different axes. We have the X and the Y axis. So the x-axis is the forward scatter, which is the size, and the y-axis is the side scatter, which is the granularity. These are color-coded populations, so blue is lymphocytes, green are monocytes, red are granulocytes, and yellow are platelets. Any other overlays could be specific marker expressions like CD3, CD4, or even CD8. Then we have the gating strategy. This is where specific gates are drawn to isolate subpopulations based on scatter and marker expression. The inset histograms can show marker distribution across the gated population. So just some takeaways for flow cytometry. This allows simultaneous multi-parameter analysis of single cells. It plays a critical role in clinical diagnostics, research, and therapy monitoring. Proper sample preparation, antibody selection, and gating are essential for accurate results.